So the the imaging system used for acquisition of images is from Speaking. It's composed, as you can see here, in 1 and 2 CCD camera and spectrograph from Speaking FX17. And in 3, it is the illumination two halogen lamps, and in 4 is translation lab scanner and sample holder. All of this equipment are controlled by lab Limo scanner software in 5. And I would like to show you a video of acquisition of hyperspectral images here. As you can see, the white reference and the samples are placed into a translation stage, and the acquisition begins by the dark image, by turning off of the lens here. And then the image of the white reference is recorded. In the last, the image of the target samples here is recorded. Of course, the type of acquisition is push broom, which means that the acquisition is line by line from the right to the left. And when the scanner moves, a new line is acquired. For this study, we used the young tuber that we bought at the market at Montpellier. These tubers were peeled, washed, and then cut into 2 longitudinal pieces. We use this tool pieces.
Ici, la visioconférence là.
Sorry?
No, go ahead, Karima.
Okay, these 2 pieces are used for acquisition of the image before drying at T0, and then the acquisition at 1 hour, 2 hours, 3 hours, and 20 hours after oven drying at 105 degrees Celsius. And then we got 5 hyperspectral images in an higher range from 900 to 1700 nanometer. These 5 hyperspectral images were corrected. That means that the reflectance images are calculated from the origin hyperspectral images, dark image and dark and white image, by using this formula. When the next step is when we acquired our hyperspectral images, the next step is the selection of the region of the interest. You need firstly, you need to know that the hyperspectral image has the cube shape. Here X and Y are spatial dimension, and Z here is the spectral dimension. It contains 224 wavelength from 900 to 1700 nanometers. We used only one image at 1230 nanometers to select the region of the interest by the simple segmentation, and the result and the binary mask is created. As you can see, this is the binary mask. in white you have the region of the interest, and in black you have no interest in the region. Of course, of each image we apply the corresponding mask to select the region of the interest. To be able to apply the chemometrics method to our hyperspectral images after select the region of interest, we need the image cube here, named here hypercube image, is folding into two-dimension matrix, two-dimension spectral matrix. Each pixel here, each pixel of hypercube image represents one spectra along all the range. As you can see here, this is one spectra, and each plane of this image represents one image acquired at one wavelength. The unfolding of hyperspectral image is carried out in the following way. Each plane is unfolding to the one column in the matrix, in the spectral matrix. For example, for the first plane of hyperspectral image, the first line of this plane is unfolded in the part of the column of the first column of the matrix, and the second line is placed below the first line, and so on, and so on. When we got our spectral matrix, we can now apply multivariate analysis or chemometrics analysis. In our case, we apply the simplest one, which is the principal component analysis. To do that, we select the first 1000 spectra randomly per image. As I said, we have five images. We got five images, hyperspectral, making hyperspectral image, making a total of 5,000 spectra. These spectra were pre-processed with normalization SMV and the second derivative. PCA will reduce the origin dimensions, which is variable and or wavelength, and will find a new few variables named sample component. And this principal component are orthogonal between them. That is mean that the first component will explain will explain maximum of the variability in the data, and the second one will explain that the variability that are not explained but by the first one, and so on. As you can see here, PCA, we decompose origin spectra to two matrices. The first one is the score matrix, T. A here is the number of new variables, principal component, and N is the number of spectra. Score matrix contain the weighted sums of the origin variables without significant loss of useful information. And the second matrix resulted from PCA is the matrix loading matrix. The loading matrix contain important variables that are responsible for the specific features appeared in the data. So here in our study, we are interested only in the first loading of the first component, because it explains the maximum of the variability and contain the weight of the all variable. Our all variable is the wavelength, the first wavelength from 900 to 1700 nanometers in the new dimension PC1. The first component, this component explains 89% of variability. Here in this loading plot, we see two strong positive bands, and at 1159 and 1400 nanometers. Where these bands are due to the presence of free water in the samples.
Karima, you have 5 minutes left.
Okay, all right. And in order to visualize water distribution in YAM during oven drying, we project all pixels of each image on PC1 axis, because PC1 axis explains the maximum of variability, and we have seen that it explains the variability of free water. And when we obtain a column vector with the score value of each pixel, this vector is folding into a plane, and this plane is the image score with the false color. Here we see the image score here in this figure. The image score obtained from T0 before drying, 1 hour, 2 hours, 3 hours, and 20 hours after drying. Dark red here represent high water content, and dark blue represent low water content. We see here that when the drying time increase, the drying part in blue evolves from the outside to the center, and the weight part in the red, they totally disappear at 20 hours after drying. We see also at 2 hours here after drying, an heterogeneous increase of dried part in blue. And in 3 hours after drying here, dried part is more than wet part. And the remaining part, the remaining wet part is more concentrated at the fibers level. We did the same work on cassava, on fresh cassava, and we just acquired the images at T0 before drying, 30 minutes after drying, 1 hour, 2 hours, and 3 hours after drying. For cassava, we see that the drying is done gradually but faster than in the yam. After 2 hours here, at 2 hours of drying, the wet part in red is nearly disappeared, except at the fiber level here. We see a little few content of water, and the water that is mean that the water in fiber level leaves slowly, more slowly.
Celui qui...
For conclusion and perspective, that this study allows us that hyperspectral imaging could be used as faster and more destructive method to visualize water distribution in yam and cassava. During drying, we have seen also that during drying in fiber level, water is more fixed and leaves slowly. For perspective, we will use variables selection to select the most informative wavelength and reduce the size, the big size of data, for hyperspectral data. Use hyperspectral imaging to measure breadth of interest in RTB foods projects, such as oxidation, color, size, shape, weight, fibers. We can also use HSI to measure physical parameters such as texture, firmness, cooking degrees, biochemical content, water content, dry matter, carotenoid, protein, starch, and then also disease and default. For more information about HSI applied to RTB crops and products, you can find the SOC that I wrote on hyperspectral imaging applied to RTB crop and product in the RTB Foods platform by using this path. And thank you for your attention.
Thank you, Karima. Thanks for this presentation. Okay, so now you can ask questions to Karima if you have some. If you have some questions, you can raise your hand using the device raised hand. I will address first a question from Gerard. Gerard is asking you, Karima, what's the duration of the acquisition of the images on the equipment? So can you answer this question? Duration of acquisition. Karima?
Yes, I don't, sorry. Just 1 minute. Can you repeat your question? I don't understand.
En français, c'est la durée d'acquisition des images.
Okay. La durée, the time of the acquisition, it's maximum 1 minute. 1 minute.
Okay, if it is 1 minute, Karima, I'm sorry, I am Gerard Ngo talking from Cameroon. Yeah, if it is 1 minute to acquire the image, therefore, how many samples can you evaluate per day?
In my case, I use just 2 pieces of fresh yam, and I have sample holders of dimension 40 centimeters per 20 centimeters. So can it depends of the size of the samples, you know, and in my case, 30, for example, 30 seconds per image.
Yes, but how many samples can you evaluate per day? That is, can you evaluate about 30 clones, or let me say 30 samples, I mean, 30 samples or 20 or 10 or 5? That's what I want to know.
In one image?
No, for the whole day. Because you said an image, it is about a minute, right?
Yeah.
Yeah. So how many samples can you evaluate per day?
I can evaluate a lot of samples with HSI. I can acquire image after image. No problem.
Okay. Thank you.
Maybe one question. Thierry speaking here from Colombia. The question might be not only how long does it take to take an image, but how long does it take to process the data to produce the useful image?
Yes, the data acquisition is easy. It's very easy, but the processing of data is difficult. You know, so it takes more time than acquisition of images.
Yeah. So how many?
How many time? Okay, for this for example, for this one, I took half of the day to get these results, because you have many steps to follow to reach these results. You know, I explained that in.
Yeah.
But now we see that some software easy to use, easy to comment dirais-je, utiliser. Well, easy to use software are in the market. If you want to put software easy to use, you can. But me, I used MATLAB. And MATLAB, you need to develop programming and something like that.
Okay. Yeah. Thomas raised his hand. So
hello.
Please.
Okay. Hello, Thomas.
Hello. Good morning from Peru. This is Thomas. In your first slide, you showed that you are peeling the roots. Where is the need to
peel the roots, and
where is the need to peel the roots to do this analysis? I don't see the need to peel it. It might be easier without peeling. Have you tried that?
We can use the wool yam tuber without peel, but I decided to peel because I don't need the information from the peel. That is why that I peel the samples.
Yeah, but it would not be necessary when you go later for starch and for protein or whatever you showed there. But, I would skip this step.
Yeah,
and don't peel it.
You can skip this step.
Okay, thank you.
Thank you.
Okay, there are other written questions from, I think, the first one from Abdul Hakim from CRI, if I'm not mistaken. Abdul Hakim, do you want to ask a question to Karima?
Yes, from IIT.
Oh, okay, I apologize.
Yeah.
Yeah. What is your question? Hello.
I think his question is, can you expatiate if you can use HSI to measure cooking degree?
Yes.
Can you expatiate, please? Use HSI to, determine the cooking degree.
Yes, if for example, you will cooking degree of boiled yam or cassava, you boil your samples and you take an image during boiling, and we can detect the cooking time, the optimum cooking time of your sample. You acquire an image for example, each 10 minutes of cooking, and you will did the same thing, and we can see the difference between the samples cooked at 10 minutes, 20 minutes, 30 minutes, and then you can detect the optimum cooking time.
Okay, so we have a question from Didier. Didier, can you please ask a question?
Yes, Karima, I have two questions.
Yes, Didier.
Yes, what about the form and the size of the sample? Do you think that is could have any importance of the collected data or not?
No, you not, the form, no, it's not important. You can use the circle, the polygon, and each shape
Okay.
you want.
Okay, thank you. My second question
Important.
is, do you think that this technology can be used to appreciate other other biochemical compounds?
Yes.
Other than water, if maybe after calibration?
Yeah. You use also this technology is used to predict several components.
Okay, thank you very much.
Thank you.
Okay, Dominique, you have a question, then Thomas again.
Yes, my question is also about calibration. How you will do to calibrate a component as phenol oxidase, for example, and distribution? How you will realize this calibration to predict phenol oxidase, for example? How can we do that?
Okay. Yes, I did. We can, for example, yes, you take say many samples at different concentration of your target component, and you take an image, and then you correspond this image to your reference value of a reference value. Each image you need to have of each samples the reference value from laboratory. You can make a calibration easily if you have that.
Thank you.
Okay, Emmanuel, you have a question? I'm not sure who is Emmanuel. Emmanuel Alamo?
Emmanuel Alamo, yeah?
Okay, go ahead.
Okay. Yes, thank you, Karima, for the good presentation. My question is, what parameters have you used this HSI to measure what you have achieved so far, apart from this measuring the water distribution?
Yes, you can, because I take just water, because the first component of the PCA explain water, and the second component, of course, that will not explain water. It will explain another component. We use the component to explain you use the component that is explain your component. For example, here in loading plot, I see that I have a positive band, a strong positive band of water. But I took PC2, principal component 2, that is real different, because it is another component. This is just for principal component. Of course, you can use or another chemometrics method to detect your another composition. If you want. Don't know if I answer.
Alamo, I think that Karima is just beginning, just beginning with the equipment. The idea is to calibrate some components on Cassava, yam,
yes, it's the same thing.
And she planned to go to different lab because it's a portable hyperspectral camera to do this calibration. But with the lockdown, it's a bit complicated at the moment, and she's doing some experiments to try the equipment. but in the future, we will have many components with good calibration.
Let me maybe add, Dominique, for example, when you go into starch analysis, starch distribution in the tuber, maybe in potato and sweet potato, you can see if the genotype is good for French fries production, because it's the starch needs to be very well distributed in the roots and in the tubers to get a good French fry. That is an application for this technique, for example.
This is just an example, of course. In
yes, yes.
Another food, many components are characterized by HSI. You know, this is just the first test on RTB products. We can test other things and see. We can characterize another component on RTB crops and products.
Yes, it's okay. Why I just asked that, we are still at the proof of concept stage. And many people that is listening to us will be thinking that maybe in addition to what we are presented, we can present some results. So that's why I just asked that question. For me, I know quite all right that we are still at the proof of concept stage, but it's for the purpose of as many that have just joined. That's why I asked that question, that we need to prove beyond measuring water, at least that equipment can measure, in RTB component can measure all other parameters that are important. Yeah, thank you.
Thank you.
Okay, thanks, Emmanuel, for this precision indeed. It is useful, I think. Any other questions? Or there is one from Claire Hershey, but I think he's not with us anymore. He's asking, Karima, you call it a non-destructive method. What is your definition of non-destructive?
Non-destructive because we don't need to prepare to sample of the sample preparation, and non-destructive. We have just to cut onto two pieces and take data and take image. This is non-destructive, like NIR and other methods.
Well, let me make a comment. When we work with grains and we work on the whole grains of maize or wheat or whatever, and then it's non-destructive. Yes. But in roots and tubers, we usually have to destroy the
yes, we can use, if you want, for example, to detect defaults on disease in the tubers. You don't need to cut or to peel or to use the whole tubers. No need to.
Okay.
It depends what you want to characterize. You know, here I want to characterize the distributional structure in the surface of the yam. I have to cut, but in other case, maybe you don't need, you know.
Okay.
A question from Julien. Julien,
and you can, if you take image, you can use your these samples to the other analysis. To the other, for example, in the laboratory analysis destructive method, we can reuse your samples, and here you can use it for the other analysis.
Okay, sorry. So question from Julien. Julien, do you want to ask your question, please?
Yes, thanks, Karima. Karima, do you have an idea of the depth of the measurement signal?
An idea of
the depth, la profondeur.
Ah, okay. Is from 3 millimeters to 5 millimeters. No. Yes, this one. The depth is not very good because it's worked the same thing. It's an NIR, the sample NIR, the same way. So the depth is from 3 millimeters to 5 millimeters. It depends of the compactor of your samples. If you have, for example, transparent samples, the dip it will be high than if you have a compact samples.
Okay, thank you, Karima.
Okay, thank you.
Any other question for Karima? No? Okay. So I think we will stop here, Karima.
Okay, thank you.
Yes, thanks for the presentation. And so we will upload the recording on our Slack platform, as I'm usually doing.
Okay.
A lot of people saying that it was a good presentation. So thanks a lot.
Okay, thank you.